Hello, good afternoon, viewers at home. My name remains Olatunde Michael, Omega Bio-Edutainment educational series lecturer. Today again, we have come active online again to talk about something very, very important, and that is nothing but to actually look at what is a mechanism, the molecular mechanisms, the effects, and what is a mutation. Don't forget that in the previous lectures, I told us about what mutation is and how important is DNA to man and to any organism. And why are we so interested in this type of topic, and why is it that whatever happens in of that, why are we so interested? Don't forget that I told us that the first thing is that the DNA is the hereditary material, and whatever happens to the hereditary material means that it can be carried from the present generation to the next generation, and because it is nothing but genetic material, so we ought to show a kind of interest in something like that. And number two is just that don't forget the rule of complementarity that has to do with knowing the composition of DNA. I told us that DNA contains, which is deoxyribonucleic acid, actually contains four different nucleotide bases. The first one is thiamine. The first one is thiamine. The second one is adenine. And triply, you just discover that according to the Chargaff rule of base pairing, C will always bind with G, and that is going to be in formation of a 3 bond. G into C is going to be 3 bond, whereas A into T is going to be 2 bond and 2 bond only. So you need to actually know what is the rule of complementarity. Okay, so I told us that today our major focus is going to understanding the effects, the mechanism of action, as well as what is going to be the molecular basis of mutation. Mutation is nothing but a sudden change in the genetic makeup. That is, whenever anything happens to DNA, that means it is going to. So, as I was saying behind the camera, that C will always bond with G, and A will doubly bond with C. And what is going to happen is that if you know the sequence on one strand, remember that DNA is double stranded molecule. So if you have CATGC, you can easily predict what is going to be on the other strand. This is going to be G, this is going to be T, this is going to be A, this is going to be C, and this is going to be G always. And just like I said, this is going to be triple bonded, this is going to be double bonded, and this is going to be double, this is going to be triple, and this is also going to be triple. This is what is called the rule of base pairing. And whatever happens at this level will go a long way as affecting the next generation, which might be first, second, or third generation. So that is the reason why we have to be very, very careful in what happens. And I have given you 3 reasons why we must show interest. Don't forget that DNA must continually undergo the normal spontaneous process, which is called replication. Don't forget that this is exactly what happens when the cell when the cell divide, this is exactly what happened. So I want to remind you of what is called the cell cycle. So it's going to be like this, and between here and here is going to, so this place, from this place down to this level, it is called the mitosis. That is the time that cell division will take place. So immediately cell divides, is going to enter a phase which is called interphase, which is the resting phase. They call it resting phase because that is when the cell will be resting and start gathering enough energy to make sure that it is nothing is going to happen whenever they want to undergo a kind of mitotic division or any division at all. Don't forget that if the cell does not, if cell do not divide, that means it is not going to obey the law of replication. So it is at this interface level that the DNA preparatory to cell division is going to replicate. So during that process of replication, if anything now happens, and what is going to happen is that there could be what is called mutation, which could be as a result of a sudden change, unprepared for. It can be spontaneous, it can be as a result of, it can be caused by chemicals, it can be caused by anything. So whenever you now have a change in one nucleotide base, or one nucleotide sequence, it could be in form of addition, that is insertion. It could be in form of deletion. Then it could be in form of substitution. So we are going to know all this as we proceed in this lecture. And what I'm actually very much interested in today is to actually know the molecular basis. We are going to cite examples. We are going to look at different types of mutation, because I don't want us to waste time any longer. So we are going to be doing it simultaneously. I will tell you, because if I should start listing the different types of mutation for you now, and by the time we now try to look at the effect later on, it might actually waste time. But at this level, I want you to understand that mutation is a sudden change in the genetic makeup. It could be at DNA. If it happens at DNA level, one of the reasons why we must show interest that DNA must continue. You know, this one is a natural way of undergoing. The cell must continue to divide. And whenever the cell divide, which is natural, that cell must make sure that it maintains its integrity. So the integrity of DNA is non-negotiable. So because if there is infidelity, or what is called inconsistency, for example, now if at the end of the day you have 23 pairs of chromosomes that is undergoing cell division, and at the end of the day you now have 22, you know exactly what has happened. Even if you have 22.9, that means the integrity of DNA is not actually being preserved. And one other thing is that any slight change in the genetic makeup or in DNA can be fatal. It can be fatal. For example, the issue of sickle cell anemia. Sickle cell anemia has to do with just the replacement of one nucleotide base. That is exactly what makes sickle cell anemia to be like that. So it is fatal. It is very, very fatal. And apart from trying to maintain the integrity of DNA, you know, mutation is the first stage. Mutation is the primary stage. It is the primary stage and very, very compulsory. You see, I cannot be here today teaching you to be your lecturer if I did not attend primary school. It is at the primary school that we start learning the alphabet. So if mutation does not take place, mutation is the primary stage for cancer. So that means cancer cannot just develop like that. It cannot. So if it does not, so it must start at the level of mutation. And that is why there are major enzymes, there are particular enzymes that have the job of performing the role of conducting surveillance, of making sure that the fidelity and the integrity of DNA is actually being maintained, to make sure that there is DNA repair, to make sure that, so an example of that is P53. P53. Any cell that has no P53 means that that cell is very prone to cancer. Apart from being very prone to cancer, that cell will undergo a premature death, or what we call suicide. That is apoptosis. Okay, that is so you can now see, and there are some mutational changes that at the end of the day, is going to lead to incorrect, is going to lead to stillbirths. So that is the reason why that at the level of DNA, we must continue to make sure that we show interest, that if anything happens, we must make it corrected. Okay, so it is very, very important. Then don't forget that I told you that in this lesson, on this lecture, I'm going to give you 3 tasks, and the 3 tasks that must be submitted just the same way I have said it. The first task is going to be that you have to list all the components, of DNA and RNA. You have to list the components of DNA and RNA. So that is A of task 1. Then task 2, still under task 1. B, you have to write what you understand by base pairing. What do you understand by base pairing role? Base pairing role in the DNA. What do you understand by that? Okay, so let's now continue our discussion. Let's continue our discussion. So in between, I'm going to give you the second task, and as well, I will give you the third task as we are proceeding. Okay, now let us start by picking those mutations one after the other, and we will look at what are the molecular basis of all these mutations. What is the molecular implication of all this? So the first one we are going to look at is a point mutation. Point, from the word point mutation. So point mutation means that there are just a replacement, or there is a substitution of just one base. So let's consider a case substitution of only one base. Let us see what is going to a scenario like that. All these are going to be hypothetical situation. So you can also imagine any kind of example that you can actually think of. So the first one we are going to consider is like when we have a copy of this DNA. Let's say, don't forget 5 prime. So the transcription or transcription will always, or replication will start from 5 prime. So let's say we have ATG, we have a CTT, then let's say we have a GA. Okay, don't forget that a video has been devoted to be able to understand the short way to understand what is called how we can, for genetic code, how can you master genetic code. So if a video has been devoted for that. Okay, now this is the original sequence. Now let's now imagine that if this original sequence, don't forget that we have what is called triplet codon. That is the amino acid, the sequence will occur in 3, and is going to code for a simple I mean a single amino acid. Okay, although there are times that you have three different three different triplet codon that are coding for more than one. So, and that is redundancy. You will know that when you go back to your considering what is called genetic code. Okay, now this is going to be 1, 2 amino acid 1, 3, 4. So let us now imagine that I've told you the essence of an enzyme or amino or a protein that is called P53. Let us imagine that this is a set of protein that will eventually code for P53. Any cell that does not contain P53 is is very, very susceptible or very prone, very vulnerable to what? So it can undergo what is called self-destruction. So it can also undergo cancer. Experimentally, it has been found that if any cell is lacking any of these, so that is the person is very prone to cancer. Okay, now let us now imagine that this is a mutated sequence. Now mutated sequence. Okay, so still from 5' end, and this is amino acid 1, amino acid 2, amino acid 3, and amino acid 4. Normally, the cell protein synthesis must start with ATG. Okay, must start with ATG, which is so this is going to code for methionine. Okay, and CTT is going to code for proline. Then the third one is going to code for GLY. Okay, and the last one, which is TAA, is going to stop. That is, whenever it has gotten to this, it's going to stop. Now let us now imagine that there is a problem of substitution of one base. So here, let us now imagine that we now have substitution of the T. Okay, let's say this one, sorry, this is a CCT. Okay, CCT. So let's now say there is now a substitution of the C with T, so that you are going to now have 5' ATG, then you now have C, which is this one, T, which is this one. You now have a T. Substitution, that is substitute, you add, then you remove. You add by removing. Substitution. So that is ATG, CCT, and this one continue, which is GGA and the TAA. What is going to the amino acid that you are going to, the resulting amino acid will now still be methionine. So instead of this one to be proline, it's going to be leucine, and glycine, just like we have it, glycine and stop. So you can now see that a replacement of just one substitution of one amino acid by replacing C with T has resulted into a different amino acid, and structurally. So let's now see what's going to be the effect of this. The effect of this is that the structure of that protein, the structure of P53, which is the hypothetical example in this case, is going to be badly affected. Now, if for example the normal structure ought to be this, and now because of the replacement of one nucleotide basis that has resulted into proline, leucine instead of proline. So it's not going to be like this. So because the structure is distorted, it is not going to perform the exact function as this one is going to perform. And don't forget the function of 353. That means the organism in which there is substitution of one nucleotide base is going to be badly affected, and the function, because whatever affects the structure is going to affect the function as well. So that is the first molecular mechanism, the molecular mechanism of substitution of only one amino acid, of only one base that has resulted into a different amino acid. Instead of us having proline, we now have a leucine. So that is the reason why it is very, very important that we consider this. Okay. Yes. Just like you see it on the board, just like you see it on the screen. Okay. You have point mutation, which is substitution of one amino acid. Substitution of one amino acid. Don't forget that when we say substitution, that is you remove and you replace. That is a substitution. You remove and you replace. By the time this one remove C, it has replaced it with another one, which is a T, and that is exactly what made that altered the structure, the primary structure of that amino, of that protein, which is polypeptide. And don't forget that primary school is so important that if the foundation is bad, if there is problems with the foundation, that means that problem is going to persist and going to persist for a very long time. And that is exactly what has happened in this hypothetical example. You can also think of any other hypothetical example to be able to explain what you mean by substitution, by point mutation. Don't forget the meaning of point. Point, that is, there is problem in one nucleotide base, just one. Replacement, it could be in form of replacement, it could be in form of addition, which is insertion. It could also be in form of deletion. Okay, that is what is called point mutation. Point mutation. And whenever we now have point mutation like this, it is very, very easy for the cell to be able to repair it. That is why after each cycle of replication, after each cycle of cell division, the cell will now enter and go again into the state of interphase. And what is interphase? Interphase actually means resting phase. That is so the cell is going to rest. It is just like the normal thing that after your daily work, after you must have overstretched yourself, you continue. The next thing is for you to be able to sleep, to be able to rest. And when you sleep, you are not sleeping. It's not that your body is not going to be active. Your body will continue to be very, very active. But the only thing is that it is going to go back, rejuvenate, re-energize, and reinvigorate. So that is, the cell will also need to do. So during the process of this cell division, before the cell will undergo another round of division or another round of replication, there are some enzymes. There are some enzymes that are actually, their primary function is to make sure that they start to conduct what is called check. They will have, what they will do, they will have the normal one. They will have the normal DNA sequence like this. They will hold it in one hand. And the newly synthesized one, they will hold it in another hand. So they will start checking it base by base to see if there is any error. So if there is any error, the cell has the ability to be able to repair that error. But if the cell cannot actually repair it at the point of mutation, that is exactly what is going to happen, that is going to result into a lot of issue before you now have the onset of cancer, onset of tumor. So a lot of things will happen. There are some cells that will contain that will commit uh, suicide. That is what is called apoptosis. There are some that prematurely they will die. That is necrosis. So all these happens, and there are a lot of things that can actually cause point mutation, including chemicals, including certain chemicals, certain food, especially all this synthetic food, can result as just a replacement, substitution of one. So if just a replacement in one nucleotide base can be so fatal to the level that you have the difference between normal hemoglobin and the sickle cell anemia. That is just replacement of just one nucleotide that has resulted into that. So that when the normal lifespan of a red blood cell, it should be 120 days. But in sickle cell anemia, it is between 60 and 90 days. So that is exactly what. So you can see how fatal, how risky that it could actually result into whenever we have what is called whenever we have mutation and it is not controlled. So let's now check. Let's go to the second hypothetical example. The second hypothetical example, let's look at the molecular basis of that. And let us see. So before I go on, let me give you, okay, it's not okay. Let me give you the second task. The second task is you have to give a molecular scenario, okay, of one when there is a mutation called frameshift, okay. When there is a mutation you can call silent mutation. When there is a mutation you can call mean-sense mutation. So you have to give the molecular scenario, that is, you have to give hypothetical example of that. That's going to be the second task. Okay, now let's go to the second hypothetical example that we have here, okay, and that is let's imagine that we have 5 prime, which is ATG. We have GCT, we have AAC. Let's say we have AAC. Let us assume we also have a GGT, okay, and we now have TAG at 3 prime end, okay. That is an hypothetical example. Now let us now see that this one is coding for this one is we are considering a protein that helps in cellular contraction, of the heart. So under normal circumstances, heart is very, very important let me say, for any human, very, very important, because that is where blood is being pumped and circulated to the other parts of the body. So if there is, so that means the heart must continue to expand and also continue to contrast. So if we now have this set of amino acid that is a polypeptide that will eventually give rise to a protein that will help in the cellular contraction of the heart. That means if there is problem with this, protein, it is either, that means the this cell will not be able to contrast any longer. And you know the implication of that. Okay, so let us say that that is an hypothetical example, and let's see what this one is coding for. This is going to be for methylene. Okay, that's the scenario. This is going to code for aniline. This is going to code for asparagine. This is also going to code for glycine. Okay, and the TAG will be for stop, to be able to stop. Okay, so that is the normal thing. Now let us now imagine in an hypothetical example. That hypothetical example, now let us now see that there is now insertion. If there is now an insertion of adenine, which is A, is inserted between the second and third codon. This is first codon, second, third, and fourth and fifth. Okay, if there is now an insertion, and if that insertion, instead of us to have ATG, okay, that is mutation occurs. Mutation occurs between this is going to be between this one, and the mutation that now occurs is that there is addition of A, insertion of A, insertion of A. Not, this is not substitution, insert. That is, you had addition. So it's now going to be, this is now going to be ATG. Because you have added A, it's going to be CAG. This T will go and meet this because it has to occur in triplet codon. Okay, CAT, CAG, then we have TAA, then we have CGG, then we now have TTA, then you have G on its own. Okay, and by the time you now look at this, you now discover that insertion of just A has caused a shift. It has caused a shift in the frame shift. That is what they call frame shift, and that is an example of what we have when we say there is a there is addition, insertion of one nucleotide. So because it is only this one that is going to maintain, it's only this one that is going to maintain the original ATG. You can see this one, there will be changes in CAG. Here too, not the same as this. This one's not the same thing as this. You can see that this one also not the same as this. And just like I told you that if this protein happen to be the protein that will help our heart to be able to contrast, at the end of the day, that means the heart will not be able to contract. And you know the implication of that. So that is the effect. So, and whatever, the structure will be badly affected. In fact, this error is even going to cost the cell a lot of things, because just one error, which is addition of just amino acid, you see now it has entirely changed the face of this protein. So instead of the protein was to be like this before, if that protein was to be like this before, now we are going to see a lot of distortion that will be too different from the original, and you know the implication. So that is why that is another scenario whereby we have insertion or addition that is going to lead to a shift in the nucleotide sequence, and you can see that whenever you now add amino, just one adenine, you see that the whole is going to shift. Yeah, you now have ATG is this, CG, so A is there, so that's why you have CAG. This one that is T will now shift to this side. That is what is called frameshift, which is TAA, which is this one, CGG, which is this one, then TTA, which is this one, and one nucleotide will now be left behind. That is not going to be, is going to be redundant. Going to be redundant. So that is exactly what happens when we talk about the hypothetical example that we have in protein that is being distorted as a result of just addition of one nucleotide sequence. And you know, whatever affects the structure, I told you that it is going to affect the whole cell. So let's now consider the third scenario or the third example. And this third example now, the first one is substitution of one. So that one is called point mutation, which is by substitution. The second one is a frameshift mutation, and I've explained the hypothetical example of what is called the frameshift for you. So let's now look at the third example, which is going to be a kind of a mutation that is as a result of deletion. That is, we'll just remove one nucleotide sequence and let us see what is going to happen. Okay, let's see what is going to happen. Now, let us imagine an hypothetical example. Let's imagine the hypothetical example where you have this case. Let's say you have a hypothetical example which is 5 prime, which is ATG. Okay, this is a GAG, this is a TCT, and this is a ACT. That's the, let's say that's the normal protein. So this sequence, this is going to be methionine. Normally, this is going to be glutamic acid. This is going to be serine, and the last one is going to be theoline. Now, if there is now a deletion, where the just a deletion, if there is a deletion that, okay, let's say we have 5 prime ATG. And now, if there is now a deletion, okay, let's assume there is a deletion of G. So this G, let's say there is now, we now make something like this. So this part is deleted already. So that this one is now going to be ATG. Is now going to be, just in this one, no, it's now going to be EAG, and this is going to be TCT, and this is going to be ACT. So immediately the messenger RNA encounters this, is going to put a question mark there. Okay, this is going to be methylene, because there's nothing like EAG, there's nothing like that. So it's either this one, this type of protein terminates immediately, or this one will just jump this, the whole of this, and go and meet this. So whenever there is any error like that, that is also called frameshift. That is also called frameshift mutation. Because the reading frameshift mutation in which the reading frame is altered. So, because don't forget, you need to go and read how protein translation again. See how protein is being synthesized, and immediately the transfer RNA now gets into this level, EAG. The reading frame will not be able to understand what is EAG. So, and what is going to happen is that there will be jump to the next one. So that means there is going to be a gap here. And you know what a gap could actually do? It's like in your heart. If there is a hole, small hole in the heart, you know what it means. That person, you know the implication. So that is what is going to happen. So, and the biological implication or the biological effect of this, biological effect, is that it's going to be fatal. Which can lead to neurodegenerative disorder. Neurodegenerative disorder. That is, the person will have impairments in decision making. The person will not, the brain, the central nervous system, the neurons. Let's say if this protein where there is deletion now, let's say it is a kind of neurons. If it's a protein that will code for a neuron in the brain, or a protein that will help in the transmission of impulses. So you know whatever is going to happen in the body, it must first of all be processed in the brain. So whenever there is now alteration, look at it, that has to lead with a shift, which is a kind of mutation in the reading frame, is being affected. So ATG now jumps to the next one. And sometimes this protein might just terminate prematurely. That's going to be more, that is going to be more common than maybe to even jump into the another reading frame. So you know exactly what is going to happen. So this is another form of a frameshift mutation that has to do with when the reading frame is being affected. When the reading frame is being affected, please take note of this as well. Okay. Now, the fourth scenario, we are going to look at the fourth scenario, and that is a scenario that is not going to be at the DNA level. It's going to be at a chromosomal level, when the chromosome is being affected, when there is duplication of certain gene. So it's going to be in a normal organism, the DNA sequence of a gene responsible for cell division can be like, we'll give you an example. Okay, and when we now give that example, we are going to be able to see it. Now let me try and cast your mind back. Okay, let's say this is a cell and this is the nucleus. So this is the nucleus or what we call the nuclear membrane, nucleus. Inside that nucleus, you have nucleolus. Inside the nucleus, you have nucleolus. Inside the nucleolus now, that is where you have what is called what? Chromosome. That's where you have the chromosome. That is where you have the chromosome. Now, located on that chromosome is your what? DNA. Located on the chromosome is your DNA. And this is your DNA. Now, on the segment of the DNA where you have different types of 4 nucleotide bases that you know already, inside that, certain segment is now what is called a gene. That is, if you come, segment of DNA is what is called a gene. And a certain gene, any certain gene must code for different protein. So you need to let that one be established. Now, if there is any problem at the DNA level, that is what we have been considering before. If there is a problem at DNA level, so that can be transmitted to affect the gene. And whenever the gene is affected, that one will be carried to the next generation. Now, at this level now, we are now trying to talk about chromosomal disorder. That is, instead of mutation taking place at the DNA level, it is now taking place at a chromosomal level. So that is what's going to lead us to the fourth one, which is a chromosomal mutation. Chromosomal mutation. And the most common chromosomal mutation is when there is a duplication of gene. If there is a duplication of gene, don't forget that one of the neurodegenerative disorder is when there is a repeat. For example, now if there is a repeat, okay. Now let us now cite an example. Let's say you have 5' going to ATG, okay, ATC. You now have GGC, ACG, and you have TTT. So this is going to be 3', okay. Now, if there is now a duplicate, this is a normal one. This is a normal gene at the chromosomal level. So let us now imagine. Let's imagine a situation whereby you now have a kind of duplication. So this is a gene, normal gene. So let's now say we have 5'. You now have ATC. Okay, we have GGC, we have ACG, and we have TTT. Okay, now if this one is now duplicated, so that you now have ATG again, you have GGC, you have ACG, and you have TTT. That is a duplication. Okay, so what that is going to happen is that there is going to be duplication of that gene, which is overproduction. Overproduction. And if there is overproduction of any gene, that gene is what? That cell where that one happens can lead to what is called tumor. Tumor, which is eventually getting into the level of what? To the level of cancer. Cancer, because there is duplication of certain gene. This is a gene, one, which is just one gene. So if the cell now duplicates this kind of gene that is going to lead to that, you know exactly what is going to happen. Okay, so and the biological effects or the effects of this I've told you is that tumor is very likely. Tumor is very likely. There will be overproduction, overproduction of certain protein. In human, if there is an absence of insulin, if there is absence of insulin, you know that is a type of diabetes. And now, if the insulin is still too much, you know that what that one can still lead into. So it's just like when you have, instead of you to have 23 pairs of chromosome, if there's now duplication, you now have 23 plus 1. That is, instead of, there is one of the chromosome, instead of it to be 2, that one is now 3. That's why it is. So this condition is what is called trisomy. Trisomy. And for those of us in genetics, when we get to our genetics class, we are going to see more of what is called trisomy. And trisomy is a kind of, when you have I told you we have chromosome 1 to 22, which are called autosome. Then chromosome 23 are the X, X, and XY, which determines whether we are going to be male or female. Now, when there is trisomy in particular, that means chromosome 21, instead of chromosome 21 to be 2, is now 3. So that's why they call it 23 plus 1. So that is a case that is called trisomy. And we are going to see this when we get to genetics class, because it is going to lead to overproduction of certain enzymes, of certain proteins. So which is very likely that that person develop cancer at the end of the day. Now, we are going to consider the last for today. We are going to consider the last mechanism or the last scenario for today before we now round it up for today. And that is going to be an error that is due to translocation. Error that is due to that has to do with translocation. Okay, so I want to make sure that you are able to see exactly what is on the screen, so that you'll be able to copy it and take note of that. I'm very sure that chromosomal mutation, which has to do with duplication of gene, you can equally use any hypothetical example to be able to explain this. So you should be able to see that. So in order not to waste our time, let us talk about the last type of mutation for today. Last type of mutation or scenario, hypothetical scenario that we are going to consider today. Okay, now before we now go into that hypothetical example, let me try and give you the last task, which is task 3. Don't forget that you have to submit not later than 30 minutes after this lecture. So task 3, you have to state one reason, why errors in DNA is so crucial to you as a biologist. So if you are a molecular biologist, you are going to discuss it in the perspective of a molecular biologist. If you are a microbiologist, you are going to discuss it in the area of being a molecular biologist. So, and if you are going to be microbiologist, environmental biologist, you have to also discuss it in that perspective. So the last scenario is when there is translocation, mutation. Okay, so what we call translocation mutation is when there is an exchange, of chromosomal segments. Okay, then there is exchange of a chromosomal segment. Now, let us imagine an hypothetical example. Let's say this is a chromosome, certain chromosome. Okay. So I want you to go back and know exactly what happens in meiosis. Okay. Now this is another chromosome. Don't forget the procedure or the process of meiosis, that at a point in time all the chromosomes will come to the middle, that is at metaphase 1, and they will start to undergo meiosis 1, they will start to undergo dikinesis, pachytene, dupletion, and so on, like that. Now this is chromosome. Let us imagine that this is a chromosome 1, chromosome 1. Okay. Chromosome 1. Now this is a gene on that chromosome which is called gene 1. Now on another chromosome, this is a certain gene which is gene 2. A certain gene which is a gene 2. So if there is now what is called a translocation, trans from the word trans, in between, translocation, location. So instead of now during the process of meiosis, all the chromosomes, they will come together, and by the time they come together, don't forget that the chromosome will break at certain point in time, so that later on this part of a chromosome, which is the chromatid, will go into the southern hemisphere. It's going to go into different cell. At the end of the day, this one will go to another cell at the end of the day, if you remember cell division very, very well. Now, instead of this one going to this side during the process whereby they have come together, if there's now a kind of exchange, if there is a kind of exchange whereby gene 1 comes to chromosome to where gene 2 is located, and this one also comes to here. If this one is you see, they will come together. And during the time when they come together like that, there could be what is called exchange of certain segments of that chromosome. So let us now see this is exactly what happened. So that at the end of the day now, you now have a chromosome, that is going to look like this. That is going to look like that. That whereby you are going to have the gene 1 still located here, and also gene 2 also located on this arm of chromosome. That is what is called translocation mutation, which has to do with exchange of chromosomal segments. And that is exactly what happens in the case. This is exactly what happened between chromosome 9 and chromosome 22 in human. Chromosome 9 and 22 in human will normally undergo this kind of mutation. And when there is now exchange of that chromosomal segment, it's going to lead to a condition that they call it CML, leukemia. They call it leukemia. So, and what is CML is called chronic myelogenous leukemia. That is a cancer. So when there is translocation between chromosome 22 and chromosome 9, when there is exchange of chromosomal segments, which has to do with a very big error, and this can occur naturally, and it can occur when there is a mutation. And that is the number 5th example that we can use in understanding and explaining the mechanism that is surrounding mutation. We have been able to see the first one that has to do with a point mutation. That is when there is just a problem in just one nucleotide as a result of substitution. We have seen what that one is going to be. We look at condition scenario 1, whereby you have a frame shift because there is a insertion, addition of adenine. There is a frame shift, and we have also seen the second type of frame mutation, whereby just a segment or a part of G that is now being deleted, and the resulting amino acid is not understandable. It is now incorrect. It is now incorrectly being interpreted. That is going to lead to incorrect error or incorrect copy of the reading frame. That is, we have seen exactly what that one means. And we have also seen what is called chromosomal mutation, whereby you can have duplication of gene. So in that area, in that kind of duplication, instead of gene 1 to come from, instead of the error to be from, to involve just the same one chromosome, to involve one chromosome, whereby you have gene 1 and another gene that is being duplicated. It is now gene 1 that will be duplicated on another arm. And we have seen exactly that one can lead to cancer. So, and this one too, that is as a result of translocational mutation, whereby there is exchange of chromosomal segment. We have seen that the basic example of this is when you have CML, which is a chronic myelogenous leukemia, which is a certain cancer. And that is when you have translocation, exchange of gene between chromosome 9 and chromosome 22. So that means there are other that that produce a kind of very serious mutational effects, that at the end of the day it is not going to be synonymous to this. So don't forget that what we have done today is just look at the molecular mechanism of understanding what is a mutation. Okay, that is, what are the molecular basis that we can use? What are the molecular scenario that we can actually use to be able to explain different types of mutation that can occur in human, especially, or at DNA level? We have seen that sometimes it might not be, the error might not be from the DNA. The error can come from chromosomal level, that is from chromosome level. So, and whatever happens, it is sometimes is going to result into tumor, cancer, and what have you. But at this level, you should just know that the primary position, or the primary, what you look at leukemia or cancer today must have started at a mutational level, when there's just a replacement or a substitution or a problem in just one nucleotide, and that nucleotide is now, because the nucleotide cannot actually replace itself naturally. The nature has actually blessed all organisms to be able to repair errors in their DNA. But sometimes the error might be too much, the error might be too fatal, especially when the supposed error repairer, they are also having problem. Maybe they are malfunctioning. So that means there's going to be, that error will go unnoticed. That is why sometimes it's very, very good that we'll begin to be very, very careful about what we consume, so that the error will not. And at DNA level, whatever happens at DNA, you can see, can also go to the chromosomal level, and what happens is going to be passed from parent to the offspring. The inheritance pattern might be different. It might be as an autosomal recessive, it might be autosomal dominant, it might also be in form of a mitochondrial problem, and it can also be X-linked dominant this. We have seen all this at so that's why you have different types of genetic disorder. All this will happen and will start from the elementary level, which has to do with mutation. And when there is an error at a mutational level, I said that kind of error can be fatal, can be everlasting, can result into something that will become irreparable. So that is the reason why. So don't forget that once again, you have to go back and read very well what is cell division. I told you that the essence of the cell to be able to undergo a period of dormancy or a period of resting, which is called interphase, is for that cell to be able to acquire enough energy. And it is at that level that the cell will now duplicate its DNA. Because if the cell cannot actually duplicate its DNA, so instead of you to have 23 pairs of chromosome, which is 2n, you are going to have only 1n, and that is not synonymous to life. So don't forget that I said that's going to be the level at which we are going to stop here today. The other time we are going to come and still talk about mutation will be maybe, and that one is going to be restricted to, might actually restrict that to those in genetics. The one that is going to actually concern us mostly is especially the microbiologists in gene editing. In gene editing, which is we are going to do gene editing and biotechnology. Okay, so thank you for your listening attention.